Okay, so if we go back to our example that we were using before, when we searched for FOXP2 on the NCBI portal search, what we're going to do now is, remember, we were interested in getting information on FOXP2 the FOXP2 gene in humans. So what we'll do now is we can click on the gene database, or the gene link in the search results, and what that will return to us is this page. Okay, so the first thing you'll see here is that we're searching for FOXP2, but now you'll see on the left-hand side this is now changed to gene. So now we're in the gene database. Okay, and remember, because we just searched for FOXP2, what we're going to get back now is results for FOXP2 from the gene database. But we didn't specify anything else, so we didn't say that we only wanted in humans. And where you can do that is using the Advanced Search tab. And again, this works very similar to the way that we do literature research, literature searching, and we'll deal with that in the next session. So I'll come back to that when we deal with the literature searching. But essentially, in this page, what we have is, you'll see that there's 225 results. Okay, but you'll see the first hit is for FOXP2. Gives a description of what FOXP2 full name for the gene is. So it's 4KBOXP2. And the first hit is for Homo sapiens, or human. Okay, so what you'll see is that because we only searched for FOXP2, what we've also got back are hits for FOXP2 in other organisms. So the second one is from mouse, third one is from the zebrafish, rat, etc. Okay, so for the sake of this example, we're interested in human. So what we're going to do is we're going to click on this FOXP2 name in the first column. And this is what you'll get back. So what I've done here is I've actually collapsed all the sections. So when you actually click on it, it might open up with some of the major sections already opened, and you can then expand those using the arrow on the left-hand side. But essentially, I just want to give you an overview of the type of information that you can get back. So again, this is just showing you, this is information for FOXP2 in humans. We have something called a gene ID. So this is the gene ID given to FOXP2 in the gene database at NCBI. So this is the ID particularly for the gene database at NCBI. Something important to see is they also give you an updated, the last time that this page was updated. So you'll see that this is updated on the 3rd of July 2016. So again, this relates back to when I was speaking about making sure that you're using resources that are regularly updated and accessing data from resources that are regularly updated. Okay, so then we have a number of different tabs with different information on FOXP2. So we have summary, genomic context, etc. And what I'm going to do is unpack some of these more important tabs and what information we can find in each one. So the first tab is the summary, or this first section is the summary section. And essentially this will give you a brief overview of what is FOXP2. Okay, so the first thing it says is the official symbol. And in this case, the official symbol is FOXP2. And this has been generated by a consortium that deals with ensuring that there are standardized names for all genes that have been identified. And HGNC stands for the Human Gene Nomenclature Consortium, or Committee. And essentially they deal with ensuring that gene names are standardized and consistent across different databases. If you want more information on HGNC, you can actually click on that. It will take you to the page, the entry page for FOXP2 for the HGNC. It then gives you the official full name, so it's 4KBOXP2. It then gives you a primary source, so this links back to the primary gene sequence entry links the primary gene sequence entry to at HGNC. It then gives you a list of related accession numbers for the FOXP2 gene, and I'll talk a bit more in detail about accession numbers a bit later. But essentially, this is a link to FOXP2 in the ensemble resource. So remember, I said that that is a genome-centered resource where we can get information, and then these link to additional database to entries for FOXP2 in some of the additional databases that are available. It then tells you what is the gene type. So this particular gene is protein coding. It then tells you if this gene sequence has been reviewed in RefSec. So it said it has been reviewed in RefSec. So there is a RefSec entry for this particular gene. Then tells you what organism it's from. It gives you the lineage down to the organism that it's from. It then gives you a couple of aliases. So what is this gene name? What has this gene also been called previously? So there's a number of aliases or names that names that have been used for FOXP2 as well. And then it gives you a brief summary of the gene. So what is the gene? What has it done? What does it do? Where is it expressed? What diseases is it involved in, etc. And then it tells you orthologs, have there been orthologs identified for this gene in other organisms. Okay, so that's the summary section. Now we'll have a look at the genomic region, transcripts, and products. So what this does is it gives you a graphical representation of FOXP2. So graphically showing you where this gene is found in the genome and showing you what is the structure of this gene. Okay. So the first thing we see that there is, it's basically we are looking at a region on chromosome 7, okay, on the human reference sequence 38, right? And what it's actually done is showing you a graphical representation of the FOXP2 gene. So in purple here, what you see, this is the FOXP2 gene. You'll see that the horizontal lines indicate exons, and the vertical line indicate introns. Okay. Sorry, I got that wrong. The horizontal lines indicate introns, and the vertical lines indicate exons. You'll see that there's arrows pointing to the right, which is indicating that the gene is transcribed in the forward direction. And you'll see that there's a concentration of exons here towards the end. And then what we're seeing here is we have some evidence from the different databases that there is evidence of alternative splicing for this particular gene. So we have different transcripts that are generated from the same genomic region, which vary in the number of exons, etc. And then, so this is a good place to sort of have a graphical overview, okay, of where is this gene found in the genome, which region is it on, and then what is the structure of the gene, what are the exons, what are the introns, etc. Okay. The next section is the bibliography section. So this is going to give you a list of publications and scientific journal articles that have been essentially where all the information has been generated, has been presented in NCBI, so where they sourced that information from. So these are all articles related to FOXP2 and articles that have been used to elucidate the function of the gene, the structure of the gene, etc. So it'll give you a list of different scientific articles, and then it'll also allow you then to link to PubMed with a full list of the publications linked to FOXP2. The next section I want to speak about is something called phenotypes. So essentially this section of the page will tell you if there has been any disease associated with variations in the FOXP2 gene. So the first thing that we'll see here is an associated conditions section, and we'll see that there has been a condition, a speech-language disorder that has been associated with variation or variance in FOXP2. So some of you might know this, that FOXP2 is a gene that is thought to have been involved in the development of speech in humans, or the evolution of speech in humans. And that there are variants, there is a genetic disorder where variants in FOXP2 have been associated with speech or language disorders in particular families. Another thing I want you to note here is that you'll see that there is a link to the OMUM database, which I spoke about before. So if you click on this link, this number here, it will then open up the OMIM database and give you information about this particular genetic disorder. And then you'll also be able to identify or to look at variants that have been identified and linked to this particular genetic disorder. And then there's some additional information about other clinically relevant variation that has been identified in FOXP2 and the associated diseases. So this looks at copy number variation, and then further down here I haven't shown you, but this will show you if there has been any genome-wide association studies that have been done where there have been variants linked to a particular disease, association with a particular disease in the FOXP2 gene. Then there's also the variation tab or the variation section. So this is the section where you'll be able to link out to resources telling you or giving you information about all the variants that have been identified in FOXP2, and then telling you, depending on the resource that you're looking at, will tell you information either about the frequency of those variants in different populations if it has been identified, and also looking at, again, variants that have been identified in FOXP2 that have some sort of clinical reference, clinical association or impact. So, for example, you click on ClinVar, it's going to tell you about variants that have been identified in FOXP2 and the clinical associations. You can also then click on NCBI has something called a variation viewer, which will, again, like the previous one where we saw the structure of the FOXP2 gene, so where the introns, exons, etc., if there's alternative splicing, this variation viewer will give you a similar graphical view, but then will show you where in the gene those variants are found. And then again, I spoke about variants and in relation to different populations. And for example, if you have a look at the SNP genome browser, it will tell you information about variants in FOXP2 that have been identified and the frequency of those variants across different population groups. So in the next session, we'll be having a look in more detail at the rest of these sections for the FOXP2 page that we returned results for.